Okay, so welcome to this next video in which we are discussing synchronous and asynchronous neurotransmitter release. Okay, so far what we've done is we've seen the synchronous release of neurotransmitter. We are in the process now of looking at asynchronous neurotransmitter release. However, before we can discuss asynchronous neurotransmitter release, we need to discuss where the synaptic vesicles that are actually going to fuse with the membrane in asynchronous neurotransmitter release are going to come from, because after synchronous release of neurotransmitter, all of the readily releasable vesicle pool has basically been fused with the presynaptic membrane, so you don't have any vesicles left in this readily releasable vesicle pool. So what has to happen is you have to liberate the synaptic vesicles that are in the reserve vesicle pool, which are these vesicles attached to the actin filaments by synapsin proteins. Now, so far we've discussed that when an action potential arrives, the calcium is going to enter the cell from the extracellular fluid, and this calcium is what drives synaptotagmin to fuse the docked synaptic vesicles with the presynaptic membrane, and that's what drives synchronous transmitter release. Okay, but also calcium is going to bind to the protein calmodulin and form these calcium calmodulin complexes, which are now going to activate an enzyme. Okay, and the enzyme they're going to activate has a rather fantastic name. It's known as the calcium calmodulin. So calcium calmodulin dependent kinase 2. So that's a mouthful. So this enzyme often has abbreviations. You won't often hear people fully say calcium calmodulin dependent kinase 2. Instead, it's often abbreviated to CAM for calmodulin kinase 2. So you'll often hear people refer to it as CAM kinase 2 in conversation, if you have conversations about this anyway. And in writing, what people will refer to it as is CAM K2. So when you see that written down, that means calcium calmodulin dependent kinase 2. Right, so I want to explain again a little bit about this enzyme, because again it's one of these enzymes that people aren't so familiar with. It's not protein kinase A, which everyone knows. It's one of these more niche ones. So I want to give you a little bit of insight into its structure and the fact that it goes around in oligomers of 12, basically. Okay, so let's discuss calcium calmodulin dependent kinase 2. Okay, so let's firstly draw the structure of one of them. Even though I've told you they don't go around on their own, they get 11 other buddies and form an oligomer of 12. But we'll firstly start with the structure of one of them, and then we can understand the structure of the oligomer. Okay, so the structure of one of them basically has these different portions. So it has a portion down here, which is known as the hub domain, okay? And this is the portion that's going to be involved in the formation of the oligomer. Okay, then you have a linker region, like so, and then over here you have the actual kinase portion of the enzyme, which is a serine-threonine kinase, so it adds phosphate groups to serine and threonine residues within proteins. So this is the kinase domain. Okay, now, usually the enzyme is not active. Now, how do you stop the enzyme from being active? Well, basically what happens is you have this little portion coming off the linker region, which basically has what's known as a pseudo-substrate, which sticks into the active site of the kinase domain. And I drew this little sort of indentation to represent the active site. So this represented the active site of the kinase domain of the CAM kinase 2 enzyme. And basically, you have this, what's known as an auto-inhibitory domain. So this is the auto-inhibitory domain. Okay, and it's got another name Some people will also refer to the auto-inhibitory domain as a pseudo-substrate. So this is also referred to as a pseudo-substrate because it's fitting into the active site as though it were a substrate, but it's certainly not going to undergo the reaction that the active site is trying to catalyze. So it's just going to remain bound in that active site and stop it from being able to catalyze the reaction it wants to catalyze. So here in purple is the pseudo-substrate. Okay, and we'll colour in the kinase domain in this turquoise colour here. Okay, what colour should we have the linker region in? I think we'll have the linker region in orange here. Okay, so I haven't actually labelled that up. This bit linking the kinase domain with the hub domain, this is known as the linker region. Linker region. Okay, and then over here in blue we have this hub domain. Now, this is the structure of a single CAM kinase 2 protein, a single calcium calmodulin dependent kinase 2 enzyme. Now, they do not go around like this. They go around with 11 other friends. So let me show you the structure of this CAM kinase 2 oligomer. Right, and basically what happens is they form two sets of 6, and then the two sets of 6 join together. So let me show you this. So it's effectively like a ring of them. Okay? So here's a ring, and then the two rings stack on top of one another. So the two rings of 6 CAM kinase 2 enzymes stack on top of each other. So let's have the two separate rings here. Now, let me draw the 6 different portions. So this needs to be divided into 6. So 1, 2, 3, 4, 5, 6. Okay, right. So now, basically you have this stack of 6 of these enzymes, and then underneath you have another stack of 6 of them. So overall you're going to have 12. Now these here, these are the hub domains, okay? So if I draw this enzyme out in full, here's the linker domain coming off the hub domain, and then you're going to have the kinase domain over here, okay? With the auto-inhibitory domain coming off here with the pseudo-substrate going into the active site like that. So let me colour it in the same way as I coloured it in previously. So this sort of wedge that's participating into this circular oligomerization of different wedges, this is the hub domain here. So this in blue, this is the hub domain of a single CAM kinase 2 enzyme. Here in purple is the pseudo-substrate, or the auto-inhibitory portion of that CAM kinase 2 enzyme. Here's the linker region, okay, and here's the kinase domain. So I have drawn one of these out in full, but there will be multiple of them. This is just one of them. Basically, every single one of these is a hub domain. So again, off here, you'll have another linker region with its kinase domain attached up there that's going off the page, okay, and its auto-inhibitory portion coming up here, etc. And down here off this one, again, this is another hub domain down here, and it will have a linker region coming off it, and it will be facing down the other way, basically. So the two bottoms of these enzymes are linking together. The two bottoms of the, well, yes, the two bottoms are linking together. Okay, so here is our kinase domain oriented in the other direction, and here's our auto-inhibitory domain with it, the pseudo-substrate going into this active site of the kinase domain. So again, I will colour this in. Here's the pseudo-substrate. Here in orange is the linker region, okay, and here is the kinase domain in turquoise. And then for this CAM kinase 2 enzyme, here is the hub domain here that is contributing to that bottom circle of 6. So overall you have these two circles of 6 CAM kinase 2 enzymes where the hub domains have all linked together to form this circle, and then you make two of these, twist one over 180 degrees and then stick the two together to make the whole oligomer. Is it a dodecamer? I would assume dodecamer, if my platonic solids are correct. Dodecamer, so that's 12, I believe. or if you just want to say it's a 12-membered oligomer, if my dodecamer is wrong, 12-membered oligomer. Oligo just means many, basically. Okay, so this is our oligomer of CAM kinase 2. Now, all of these enzymes in this oligomer are currently inactivated. Okay, and I just want to stress, this is how it exists in the cytoplasm. It doesn't go around on its own. It's in one of these huge, great complexes. Now, basically at the moment, all of them are inactivated. They all have their autoinhibitory domains stuck in their active sites, and that's stopping them from being able to add phosphate groups onto serine and threonine residues. However, if calcium goes up, calcium's going to bind to calmodulin to form calcium-calmodulin complexes. And what's going to happen is that the calcium-calmodulin complexes are going to come in here and bind to these autoinhibitory domains here, like so. And what that will cause is it will cause this pseudo-substrate here, or this autoinhibitory portion, to be pulled out of the active site. So let me show you one that's now been activated over here. So we'll have this other enzyme over here. And basically, here is the kinase domain here. And now we have calcium-calmodulin complexes bound, well, a single calcium-calmodulin complex bound to this autoinhibitory domain, and that means that this pseudo-substrate has been pulled out of the active site. So basically it means that the kinase domain is now capable of actually functioning because it no longer has this silly pseudo-substrate stuck in its active site. So here's the link region in orange. I'll draw the orange dots representing the calcium bound to that calcium-calmodulin complex there. Okay, and here in turquoise is this now active kinase domain. Okay, and then just to finish it, here's the hub region that contributed this this linker and then the kinase domain of this CAM kinase 2 enzyme. Okay, right, so that is how calcium-calmodulin complexes activate the CAM kinase 2 enzymes. Now, another interesting fact is that there are different isoforms of the CAM kinase 2 enzyme, and they vary in how long this linker region is. Now, if the linker region is very, very short, then you're basically going to have everything very, very dense. The kinase domains are going to be very, very close to the hub regions, and it's going to be very, very difficult for calcium-calmodulin complexes to gain access to the autoinhibitory domains so that they can bind to them and cause them to come out of the active site of the kinase domains. So basically, CAM kinase 2 isoforms or isoenzymes which have short linker regions, those are more difficult to stimulate and need higher calcium-calmodulin complex concentrations to stimulate. Whereas isoforms of CAM kinase 2 which have very long linker regions, those are far more open basically, and calcium-calmodulin complexes will find it far easier to gain access to this autoinhibitory domain here and cause it to leave the active site and activate the kinase domain. So CAM kinase 2 isoforms, which I haven't talked about. I haven't talked about the different isoforms. I've just talked about CAM kinase 2 in general. But the CAM kinase 2 isoforms, which have long linker regions, are far easier to stimulate and will be stimulated at lower concentrations of calcium-calmodulin complexes. Okay, so that's just a little off the side. Now, the important thing is that the calcium-calmodulin complexes have now activated our CAM kinase 2 enzymes. What's going to happen is that the CAM kinase 2 enzymes are going to phosphorylate those synapsin proteins, which, to remind you, are those proteins that are holding the synaptic vesicles to the actin filaments, basically, and keeping them in the reserve pool. Okay, so they're going to come in, phosphorylate these synapsins. And basically, when you phosphorylate a synapsin, so let's draw our synapsin here, and it's been phosphorylated, so it's got a phosphate group sticking off it here. Okay, when you phosphorylate a synapsin, it no longer wants to bind actin, so it's going to abandon the actin filament. So here's the actin filament drawn here. It doesn't want to bind to that anymore. So off it goes, it leaves that. And also, it doesn't want to bind to the lipid of the synaptic vesicle either. So it stops holding on to both of its cargo. So here's the synaptic vesicle with the synaptic neurotransmitter contents. And here is our actin monomers in green. Okay, so it abandons both the things it was holding on to, and that means that the synaptic vesicles are no longer attached to these actin filaments. So the synaptic vesicle is now going to go off, basically. We say that the vesicle has been liberated from the reserve pool. So this is the liberation of the reserve vesicle pool of the reserve pool. Okay, so what's going to happen is these synaptic vesicles are going to diffuse towards the presynaptic membrane. They're then going to dock, form core snare complexes again, and core snare complexes should not cause them to fuse. So now, what is actually going to cause those synaptic vesicles to fuse? Well, we'll look at that in the next video.